Welcome to the webinar on exploring sequence alignment tools with a job dispatcher. My name is Nandana Madhusudanan and I'm from the job dispatcher team. So before we proceed, I'll just turn my camera off. Okay. So as Diana mentioned, if you have any questions during the session, please feel free to type in the Q&A box so that we can answer those at the end. So okay, so this webinar will cover different sequence alignment tools available in Job Dispatcher, both pairwise and multiple sequence alignment tools, and how you can access these resources both using a web form and programmatically. I will also show you a demo as well for both these approaches and some tips for using our tools. So let's first see what is a Job Dispatcher. The Job Dispatcher service at EBI provides a free and reliable access to several sequence analysis tools. In Job Dispatcher, we have 10 categories of tools and a database entry retrieval system called dbfetch. Overall, in Job Dispatcher, we have 50 plus tools available. So the main advantages of using sequence alignment tools via Job Dispatcher is we provide the complete tool infrastructure. So you don't need to install any tool locally. We provide an interactive web form which produces a visual output. In addition to the web-based applications, we have a web services as well, which is based on RESTful and SOAP APIs. There are some built-in workflows available in some of our tools that allows you to pass the result of one tool output to another. So which I'll show you during the demo. In addition to external users like you, we have a few EBI service teams as well, like ENA, Uniprot, Ensemble Genome, where they use our web services. So in order to access our tools, you can directly go to the EBI service page, which is www.ebi.ac.uk/services. So this service page lists all our tools and data resources that are available at EBI. If you know the tool name, you can directly search for it, or you can click on the link under this programmatic access block. So I hope you can see that. Okay, which will lead you to the Job Dispatcher homepage directly. So this is the homepage for a Job Dispatcher sequence analysis tools, which is a Confluence page where you can find a brief description about our services and some YouTube video tutorials. So the tool categories that we provide are mentioned here on the left-hand side, which is again a documentation pages to our tools. In order to list our tool categories, you can go to the tools help documentation page where you can find a list of tools under each category. So this is the tool category page. There will be links available to each tool page. So we have several other categories of tools for like phylogeny, protein functional analysis, sequence similarity search, etc. For sequence alignment, we have both pairwise and multiple sequence alignment tools. Pairwise sequence alignment tools can be found under this link, like www.ebi.ac.uk/tools with a capital T/PSA, which is for pairwise sequence alignment. So these are the list of tools we provide in PSA category. There are tools available for global, local, and genomic alignment. As you know, in pairwise alignment, we try to find the regions of similarity between two query sequences. So global alignment tools helps to create an end-to-end alignment, whereas a local alignment aligns internal matching regions within sequences. In genomic alignment category, we have a GeneWise tool that compares a protein sequence to a genomic DNA sequence. allowing for introns and frame shifting errors. Now we will see how you can use a pairwise alignment tool. So I have selected Empos Needle for this demo. So let me go to the homepage for Empos Needle. So for all of our tools, there will be a brief description about the tool, and then you have option to select the type of sequence that you are providing, like protein or nucleotide. Since this is pairwise alignment, you are expected to provide 2 sequences. So you can either paste your sequences here or choose the file upload option. Your sequences should be in any of the supported format, like FASTA, GenBank, EMBL, etc. There is a documentation page available here if you click, which has a list of example sequence formats. So if documentation are available in each section, like if you have any doubts, you can just click on the Confluence, like this is a Confluence page, help and documentation page. So you can just go to that. There is more parameters available here, like if you want to choose a different a format for your result, you can choose it from here. And if you wish to use a different metrics, options are available. And you can also set values for your gap penalties. If you wish to be notified by an email, like you can click here and provide your email and job title. So once the results are available, you'll get an email. So let me just show you an example. I am using human hemoglobin a protein sequence from UniProt in FASTA format. So that's my first sequence. I've uploaded that here. And I am selecting the second sequence, which is again hemoglobin from mouse. So I'm using the default parameters, and when I submit, you can see this is validating the parameters. Once the validation is successful, you will get a job ID. So this is the job ID. This is a unique ID. So this is the alignment. So this is the pairwise sequence alignment produced by Empos Needle. So if you wish to download the alignment, you can go to the view alignment and right click, then save your alignment to your local machine. There is also a tab called submission details, which give you the version of the tool, name of the program that you used, and the launch date and end date. So when did you start the job and when it is finished, and some other details like the parameters that you provided, and the commands and the files, the output and input files. So that's all about Empos Needle. So we discussed these things. So yeah. Now coming to MSA, the landing page for MSA is this, www.ebi.ac.uk/tools with a capital T/MSA multiple sequence alignment. So there are several tools under this category. So in this WebRank tool, we only provide the web services. The web page is not managed by us, it's managed by Goldman Group. Now we will see how to use a multiple sequence alignment tool. So I've selected K-Align for the demo. So same like PSA tools, we have different sections available here as well. So since it's multiple sequence alignment tool, you are expected to provide 3 or more sequences here. you can use a file upload option, or you can paste your sequences here. When you paste your sequences, make sure that each sequence starts in a new line. You are not supposed to provide the sequences like this. Again, you have more options available if you wish to change the parameters, the default parameters. So we will just take an example. So I have a multiple sequence alignment file for hemoglobin protein from different organisms. So this is in FASTA format. I'm just submitting my job. So again, I got a job ID. So this is the multiple sequence alignment produced by K-Align. So here you can see some extra tabs for multiple sequence alignment tools. So let me explain you. Like if you would like to download the alignment file, you can right click and save, like go here and right click and save your alignment file to your local machine. Yes, like this, same like PSA. And in there is, if you wish to see the alignments in color, you can click on the show colors. So you get the colored alignment. More about the color, the indication of these colors can be found at the Bioinformatics tools FAQ documentation, like for different amino acid group, there are different colors. So that's it. If you click on the result summary, you can see all the results produced by the tool, like the input, the tool output and the input sequence that you provided, and phylogenetic tree and percentage identity metrics. So this phylogenetic tree you can again see in this tab. So this is the phylogenetic tree, which uses a neighbor joining method, and this is the tree data. This is generated actually from the cluster tool. And if you wish to pass your alignment to some other program, there is an option. So result viewers. So if you wish to view your result in JALView, JALView is a third-party tool. So if you're using it for the first time, you may need to download the JALView desktop version. So I have already installed that in my machine. So this is the link where you can download it. And once you have it, you just copy this alignment link and then open JALView. So there is in file, there is an option to add sequences from URL. There you can provide this URL and click OK. So now you can see the sequences are added to this one. These are the previous sequences. So we can remove those sequences by just right click and hide sequences. So now I have the sequences that we got from K-Align, the alignment that we got from K-Align. So if you wish to see colors, you can choose which format you want. And if you wish to save it, again, options are available to save that export image. Okay, so you can save, if it's like a PNG file, you can save it as like that. And from the alignment, you cannot save this as such. The better option is you can just take a screenshot. But in JALView, you can actually save that file image. Another option is MView, like where you can reformat your multiple sequence alignment results. So if I launch MView from here, you can see the previous job ID, K-align job ID is passing to MView. So it again reformats your multiple sequence alignment. There are options available here. Yeah, so you can submit and so this is the MView result. And if you wish to generate a phylogenetic tree, there is again option available from here, symbol to symbol phylogeny. So here again, you are passing the previous K-align job ID. So from the K-align result, you got a phylogenetic tree, which is based on neighbor joining method. So if you want to change the clustering method, you can use whichever, like UPGMA. So that's all about multiple sequence alignment. Yeah, again, the submission details, which has like same like any other tool, there are like a version and program, and the parameter details are also described in the, I mean, can get from the submission details. So we have another way of accessing our tools, that is using web services, where you are communicating with our servers programmatically. So web services uses the same approach as you do for web browsers. So whenever you are submitting a task via browser, a respective web service is used to communicate with our servers for getting the required data. So here the difference is you are avoiding the overhead of a web browser and directly talking to the web service programmatically. We are not going to any further details of this web services in this webinar, but I'll just show you how to use a web service clients. So web service clients are already written programs or scripts that calls the web service and are available in our Git repository. So you can download and use it for your analysis. So this is the Git repository for job dispatcher analysis, sequence analysis tools. So if you go here, you can see there are several web service clients available in different languages. So there is a brief description about how to use the clients. And in this demo, I'm using a web service client, Python web service client for a PSA tool. So if you see here, you can see before using, like you should make sure that you have a Python version 3.5 plus, and you need to have the dependencies XML trump to requests and requests. So if you don't have, you can install it. So I am going to the Python client and Empos Needle. So I'm opening my terminal. So I am downloading this file, this Python client for Empos Needle to my local machine using wget. So now I have Empos Needle. So if you want to get the help for this client, you just need to type the script without passing any arguments or using -- help. So if I do this, as you can see, it lists the help. So here you can see this is for pairwise sequence alignment. And the required parameters are email, sequence type, A sequence, first sequence, B sequence, second sequence. So these are the required parameters. And there are optional parameters like metrics. These are the other parameters that you already saw in the web form. So let me just show you how to run command how to run an analysis. So first you provide your required parameters. First email, then sequence type. I am giving a protein sequence, then a sequence. I already have this sequence here in my path. So then the second sequence. So once you submit, you will get a job ID. So this is your job ID. The job is finished and you got the results here. So this is your alignment file. So if you don't know, so if you want to try different parameters and you don't know what are the parameters for this particular tool, again, there are options to see the parameters. So that is here, you can see params list input parameters. So I'm just passing the params argument. Oh, sorry. So these are the parameters for this Empos Needle. If you want to know the values for a particular parameters, again, there is an option, param detail. So just use param detail, then the parameter. Now you can see the list of matrices available for Empos Needle. So that's all about using a web service client. So yeah, likewise, if you want to know the status of a particular job, you can use like minus status, then the job ID. Minus status, minus job ID, then provide the job ID. So that will get you the status, whether it's finished, running, failed. You will get the status. So if you wish to get the result, you can just use this poll job and job ID. So yeah, that's it. So there are some important notes or tips that you need to keep in mind while using our services. Your analysis results are stored in our server only for a week. So like from the job submission. So make sure that you download the alignments or the results for future reference. Suppose if you're using the web services and you want to see the result in a browser, you can use it from this link directly. So like now I have where is the file? Okay, so if I just wanted to see the result in the browser, I can just copy this job ID and then you can paste it here. It will show the result in the browser. And note that none of our tools are meant for genome synteny maps or for whole genome analysis. And if you're using our web services, there is a limit for the number of jobs. The maximum number of jobs you can run simultaneously is 30 jobs. So it is to provide a fair access to all our users. So if the parameters will be retained once you go back from the result page to the initial page. So if you are in the result page and if you go back to if you press the back button, then all the parameters will be retained. If you want to use the default, again, you need to refresh the page. It's always better to use a simple file format like FASTA. Suppose if you're using a multiple sequence alignment tool, and you have like several number of large sequences, and if you're providing it in EMBL or GenBank format, it would be like there is a maximum limit, file size limit for our tool, so it can exceed the file size limit. So it's always better to choose a simple file format. Then when you're using the MSA tools, make sure that you are choosing the right tool, like go for a global alignment tool if you are having similar sequences that are of roughly equal size. And global alignment is used to compare homologous genes or proteins with same function. And select any local alignment tool if you're interested in finding conserved regions or finding similar sequence motifs within their larger sequence context. You can go for emboss structure if you have a larger sequences and looking for global alignment. Similarly, if you have larger sequences for MSA, use K-align that concentrates on local regions, and you can use cluster omega for medium to large sequences. One more thing, the EMBU is categorized under a multiple sequence alignment tool, but it is not actually a multiple sequence alignment tool. It reformats the resultant alignment that you get from a multiple sequence alignment tool. If you have any questions or queries, you can always contact us at support@ebi.ac.uk or via the support form. If you're using our services, please cite us using this. Yeah, that's all about today's webinar. Yes, if you have any questions, please.
Thank you very much, Nandana. So we have some questions in the Q&A. If you could check the open questions. So the first one is it possible to run the tools from the command line using Bash language instead of using tools from the web? If yes, do the tools provide any documentation in order to use them from the command line?
Yes, you can actually. I think what he's asking, whether you can run. Yes, we can, because as I showed you, we have web services clients available already. So you can use that. Or if you want to integrate the web service calls into your script, yes, that is also possible.
Okay, thank you. Did it answer your question? If you want to add anything, you can add it in the chat and we'll come back to it. So the other question here is there any annotation of result obtained possible? Can you please summarize the advantage of using this software over the tools present on any CBI like BLAST and Cluster and W?
Okay, so the BLAST tool that we are using is also from the NCBI, but the difference is that in NCBI they are using their databases, whereas in EBI BLAST you can search against the databases provided by EBI. So yeah, that's an advantage.
Thank you. So the next question is how frequently the database are updated here? How many species are supported here in database? Okay, I just can't see the full question.
So Empos Needle won't have, we are not searching anything against database in Empos Needle. So these all are pairwise sequence alignment. It's not sequence similarity search. Sequence similarity search is another category.
Okay, thank you. So can you please explain gaps and plenties, your words, so that we can understand these terminologies?
Okay, so when you in an alignment, when you apply a gap, there should be a penalty given, like a um, uh, there should be a penalty. So it's a number that you are giving while calculating the alignment. So if you would like to read more about, I think there are links available in the help and documentation. If not, just contact us on support so that we can redirect you to the right pages.
Thank you. And the other question is, what are the advantages of using scripts over click and get interfaces that looks difficult to common user?
Okay, so this is mainly used when you have, so many alignments. Like if you have to perform maybe you have to perform a pairwise alignment or multiple alignments for like maybe 50 sequence or something, like, every time you have to go to the browser and click and, provide your parameters and all these things. But if you're using this, programmatic, if you are using programmatic access, it's more easy. Like you can just script it and, you can, it's more convenient in that sense. And other way is like if you're using a workflow, if you want to create your own workflow, this is helpful.
Thank you. So we have another question. What is the medium large sequence, and when would you use MAFT or MASA?
Okay. Yeah. Okay. So okay, medium large, what I meant is like sometimes I have seen like users, they have like very lengthy sequence, like DNA sequences, and which are suppose in cluster they are trying to like a very big sequence, and some sequences are like a very, it's not so very big sequences, and the number of sequences also like huge. So that's what like that's what I meant by large sequences. And yeah, MAFT and muzzle, of course you can use. Again, it depends on your requirement, which tool you want to use.
Thank you, Nandana. So the other one asks if you can please demonstrate genome alignment.
Oh, okay. So if I think we have only one sequence tool for genome alignment, which is GeneWise. Yeah. So do you want me to show that now?
Yeah, we could maybe move to the next questions, and at the end you could show that,
yeah, okay.
if that's okay. So we come back to that soon. So the next question is, are sample sequences mapped to NCBI reference genomes or other resources?
I don't think that comes under the scope of this webinar. I am not sure what the question is exactly.
Maybe who asked the question, if you want to elaborate on that.
Yes, please.
Yeah, so so we can then move to the next question. What MSA tool would be the best if I want to align sequences that have one common functional protein domain, but they vary drastically in other regions, length and structure?
That I may need to check. Like if you could again contact us on support. If you, yeah, I think I need to check that. I cannot like it's again their requirement, right? So it depends on the sequence size, and yeah, since it's protein, obviously it's not that big, but yeah.
Oh, thank you. So Anastasia, so yeah, write to the help desk, and then they might help you with that. That's okay. So we have another question. Can we use Google Colab to run alignments? Can we get a script?
That I am not sure. Like, I may need to check with my developers. It's, yeah, sorry. Yeah.
Yeah, so Obdulia, if you also could write maybe to the help desk that she shared the contact, so maybe you can get a reply later on to that. So the next question is, are sample sequences, no, this one you already said that you need some clarification, the question. So the last one, it's can we align more than one genome sequence for the comparison study? So
I don't know which tool he mentioned, because none of our tool is meant for genome sequence comparison.
Neha, do you, if you want to write which tool you are referring to, maybe it could help Nadana to answer that. So while we wait to see if we have anything added to these questions, we could move to the one that asked you to demonstrate the genome alignment, since we still have time for that.
Okay, so this is an example sequence already provided. You can just, I'm just using that example sequences. So as you can see, your protein sequence, and then paste your DNA sequences. So it compares protein sequence to a genomic DNA sequence. So then, I mean, there are options again. Then submit your job. So that's it. So you get the output. This is what they mean. Is that enough? Hello?
Yeah. Okay. Okay. And we have Neha did complement the question, the first question, I think. So I think she's asking if there is there any tool available for like to align more than one genome sequence for comparison study.
No, we don't.
So yeah, any other questions? So that's it. If you have no more questions, we will end this webinar. And yeah, thank you, Nadana, for joining us and giving this great talk.
Thank you all for joining the webinar too.